Hello, and welcome everyone to our today's webinar on first-in-man studies, strategies for a successful outcome. To be presented by Dr. Meera Desai and Dr. Kiran Mathak. I am Yuvragi Chandeli, and I represent Veeda Clinical Research, one of the largest independent CROs in India, offering its clients a wide range of comprehensive clinical services, ranging from BA/B studies, PK studies, to patient-based trials, biosimilars, biopharmaceutics, and much more. As your facilitator today, I would like to introduce you all to a couple of points before we begin this session. We will be running a live Q&A at the end of this webinar. So we have enabled our Ask a Question feature, which is right there on your screen. So if you have any questions, just write them in there. Also, this webinar is divided into two halves. The first half will be covered by Dr. Meera, and later half will be covered by Dr. Mathak. Moving on, let me introduce you all to our first presenter today, Dr. Meera Desai. Dr. Meera is a renowned clinical pharmacologist of the country and an ex-professor and head of clinical pharmacology department at BJ Medical College and Civil Hospital, Ahmedabad. She has been the chairperson of DSMB for phase 1 studies and safety adjudication committees. She's one of the finest teachers with many MD pharmacology students and PhD students trained under her. She's also an expert for CDS-CO, and she is well respected in the pharma and biotech field for clinical trials. Dr. Meera Desai currently heads the Department of Pharmacology at Nutan Medical College and Research Center. She is continuing to pursue research in clinical pharmacology, clinical trials, and research ethics and medical education. Without taking much of your time, I would like to virtually call upon Dr. Meera Desai to begin this session. Over to you, Dr. Meera Desai.
Thank you, Dr. Yuvragi. It's really a pleasure and an honor for me to be a panelist organized by Veeda Clinical Research, and I extend my sincere thanks to Dr. Mathak and Veeda Research for giving this opportunity. So let us start with the first session on the first-in-man studies, strategies for a successful outcome. And I will be mainly sharing about the basic scientific aspect of the first-in-man studies. And as we all know, that the drug development process is highly technical, complex, time-consuming, enormously expensive. And in spite of that, the success rate is very less. And quite often, the investigators, the sponsors, they are at the crossroad to face a difficult question, whether is it worth continuing the study or needs to be terminated. While the early phase clinical trials, also known as a first-in-man or proof of concept studies, they are of two types, first-in-man and first of its kind, which is a new chemical entity and never been tested in human beings. And the second one is a first-in-man, but not first of its kind. These are the important milestone and foundation of a successful clinical drug development process. And the first-in-man studies, they link the preclinical data to a potential drug candidate for subsequent therapeutic application. And these studies also allow the drug developers to decide whether to proceed further with the large, more expensive studies. Now, the objective of these studies is to determine the safety, tolerability, organ susceptibility. Secondly, the pharmacokinetics, especially the dose range and the maximum tolerated dose, and to recommend the dose and subsequent schedule for subsequent phase 2 trial, and if possible, even to estimate the pharmacodynamics. Now, historically, the first-in-man studies, they are conducted in a logical sequence. The first one to start is a single ascending dose, followed by multiple ascending dose, and subsequently the effect of food, gender, age, drug-drug interaction, and QTC interval. Now the key questions for these studies: how to select the starting safe dose? That is the fundamental question. Secondly, what is the method for dose escalation? Thirdly, how to specify the dose limiting toxicity and maximum tolerated dose? What should be the design and the number of subjects per cohort? What should be the specific study requirement? And the most important, safety monitoring of the subjects and the endpoints. Let us start these answering or discussing these questions one by one. The first question, selection of the safe dose, starting safe dose. It is a cornerstone for the first-in-man studies, and it is challenging because there is no uniform standard approach. The first dose should not be very high, which proves to be too toxic. At the same time, it should not be very low, which requires several escalation and which will prove to be very expensive. The approach should be to mitigate the risk of toxicity and to obtain pharmacological activity. Now, there are 5 different approaches. The first one, which is all over the world, which is accepted, it has been used for all these years and it has become a benchmark. That is the maximum recommended starting dose by no observed adverse effect level in the animal toxicity, specially tested in the metabolically appropriate and sensitive animals. The second is the minimal anticipated biological level, which is also known as MEBL. It is approved again by the regulators, especially the European Medicine Agency. The third approach is a pharmacokinetic and pharmacodynamic modeling, and especially for the oncology trials, especially dealing with the cytotoxic drugs. These 1/10 of the LD10 in rodents, especially in mice, and 1/6 of toxic dose low in dog or monkey, that has also been used. Now, the most common, universal, quite safe, which has been used all these years, it is a logarithm. And once you obtain the animal dose, that is converted into human equivalent dose by an algorithm, there are 5 steps, and which gives us the maximum recommended starting dose. Along with this, there is also a table of Km factor, and these factors are used in the calculation step-by-step process as per the species obtained. To simplify this calculation, let me share with you an example. Now, the NOEL determined from the toxicity studies in dogs, suppose it is 20 milligram per kg. The first thing is to convert into a body surface area by applying the Km factor of dog, that comes to 400 milligram. Now, body surface area, the whole objective is it correlates well with the human, especially obtained from the rodents or even the canines. The second step is to determine the human equivalent dose, divide by the human Km factor. And so you can see the NOEL, which is obtained from dog, it is almost reduced to half by converting into the human equivalent dose. Lastly, apply the safety factor of 10 to obtain the maximum recommended starting dose. And that comes to almost 1 milligram per kg for adult healthy male. Now, in certain circumstances, especially when you're working with the drug products which are likely to have an exaggerated pharmacological actions, or certain drugs which follow a very steep dose response curve, or the therapeutic index is very low, it is also considered to apply pharmacologically active dose, especially vasodilators, anticoagulants. These are some of the group of drugs where this is also applied. The dose which is obtained by this method, you compare it with the maximum recommended starting dose. Now, if this pharmacological active dose is less than MRSD, consider further lowering of the dose in view of the safety of the subjects. Now, this also, let me share with you that the two methods which are very commonly used across the country, across the world. So what is the fundamental difference between the Noel and Mabel? Noel, it is widely used, as I said, and it has a good safety record all these years. However, the focus is on the toxicity rather than pharmacology of the drug. And that is why it cannot be applied for drugs which are acting at a very low concentration, at a physiological level. For example, the targeted drugs, prophylactic vaccines, locally administered drugs. While in contrast, Mabel, which has been introduced after the 2006 tragedy, that was a turning point, and the focus is on the pharmacology of the drug, especially the mechanism of action, whether the substance is agonist, antagonist, the receptor affinity and occupancy. And if we compare the dose level, it is the lowest active dose rather than the highest upper end of the active dose. And it is considered to be more safe and a very conservative approach for drugs which are high risk. And this is a slide which can clear and which can bring more clarity. When in the therapeutic dose range, as you can see from 1 to 100, the initial starting dose where the therapeutic effect just starts, that is considered to be the Mabel. And that is, as the name already suggests, minimally anticipated biological effect. So it is the lowest active dose which is used. On the other hand, Novel, that is an extreme end, the highest, largest dose, especially of the active dose without producing toxicity. So both of them, they are at the extreme end. Now, it is up to the researcher who can decide, especially the toxicologist as well as the clinical pharmacologist, depends upon the study drug, depends upon the safety, depends upon also some severe toxicity, irreversible toxicity, unexplained toxicity observed in the animal studies, or even the nature of the investigational product, whether it will be, you require a very high dose to produce a pharmacological action, or the drug will be effective also at a physiological level. And that is why, as I said, there is no uniform standard. It has to be as per case-to-case basis. Now, it may happen practically, and that really happens, that when you are using different method, the estimates may be different. And this becomes a dilemma for the investigator and the sponsor, which dose should be selected. In the best interest of the safety, the lowest value should be selected. Secondly, if the preclinical data is not strong, we are not sure about the species difference, the difference in the bioavailability and the kinetics. What will happen if the same drug, which has proved to be very good in the animals, whether the same will be there in the human beings? Consider reducing the starting dose. That is a fundamental principle. Not only that, the dose, increase in the dose also should be very slower in the increment. And last but not the least, be extra cautious when you're working with the biological products. And as we know, the 2006 tragedy, which happened with the monoclonal antibodies, because we know that these substances, they can interact with the body system and they can result into severe anaphylactic reaction, cytokine storm. And that is why one has to be extra cautious when you're working with the biological products. With this, let's move to the second question of dose escalation. Is there any method? Yes. And the strong recommendation for dose escalation, which has been recommended even by the regulators and approved, is a sequential dosing, which is also known as a sentinel dosing. And this dosing is with an appropriate period of time observation at each dose level before you move to the next dose level. And there are two methods so far. One is a rule-based, traditional 3+3 design, and the second one is a model-based design. Let's have a look at it. This is a slide showing that how this traditional rule-based 3+3 design works. The first and foremost thing, once you have decided the starting safe dose, enroll 3 subjects on the starting safe dose, observe them for the required period of time for dose toxicity. It may happen that none of the subject develops toxicity. Yes, that is the step that you have to escalate to the next dose level, X+1. However, if one subject develops toxicity, enroll 3 more subjects on the same dose level. So you now have 6 subjects. Observe them for the toxicity. If one develops toxicity out of 6, still you can escalate to the next dose level. Because if you calculate the percentage, this comes to around 17%. And that is an acceptable limit as per the regulators. However, if the toxicity is observed in more than 1 out of 6, similarly, either more than 1 in 3 at the very step, that itself indicates that you have already touched or exceeded the maximum tolerated dose, and there is a time to move to de-escalate the dose to X-1. So this traditional design, it has been used for cytotoxic, non-cytotoxic drugs, and there are a number of publications on this, and it has proved to be safe, very straightforward. Importantly, it is investigator and clinician friendly because you don't have to apply, you just have to follow the rule. And it is quite adaptive because there is a provision of escalation and de-escalation of the dose. However, the endpoint is dose limiting toxicity. Moreover, there is also a scope for modification. Instead of 3+3, you can have 2+4, 3+3+3, 3+1+1, and there are a number of especially articles published with these type of studies. However, it has been observed that it is quite time consuming because it may require several escalation. At the same time, especially in oncology trial, it is likely that the patients are exposed to the subtherapeutic doses, while the objective in oncology trial that the patient should be benefited. So this is the limitation of these traditional designs. To overcome this limitation, the model-based design has come up, which is a Bayesian model-based method, and it is actually working on the dose toxicity relationship, which estimates the probability of maximum tolerated dose. However, it is more precise, very efficient, often used in oncology trials, but it is complex, requires expert biostatistician and spatial software, because at each dose level, the data is entered into the software, and based on the software, the results, the escalation, and the number of subjects enrolled is decided. Now, in both these methods, the endpoint is dose limiting toxicity. And this has to be specified before you start the study. And this dose limiting toxicity is not generic, it is protocol specific. As per the proposed therapeutic use, the dosing schedule of the study drug. And this has to define that what we really mean by dose limiting toxicity. A clinically significant adverse event or an abnormal lab value, which is unacceptable due to severity, irreversibility, or it is grade 3 or more as per standard grading criteria. This has to be specified before we start the study. Further, specify the acceptable and unacceptable limits. For example, the one which I have shown you, especially in the oncology studies, what is unacceptable? Either it is clinically irreversible severity, for example, diarrhea, vomiting, which will be very common. But if these adverse events, which cannot be controlled on the standard drug therapy, and if the proposed dose drug is to be used on the daily basis, that becomes acceptable. And this has to be specified. Moreover, enrollment in the next cohort should always be there after the review of safety data. This is very, very important. You cannot escalate more to the next higher dose before reviewing the safety data. Determine the subsequent doses as well as the maximum tolerated doses. And this is the slide showing what will be the maximum tolerated doses. As you can see, the enrollment of the subject, 3, no toxicity, move to the next dose, higher dose level, enroll 3 subjects, 1 subject develops toxicity, still within the acceptable limit, move, enroll 3 more subjects at the same dose level, 1 subject develops toxicity, move to the next. Out of that, at the higher dose level, you can see that the toxicity is 2. And this indicates, and this 2 means it is 33% or more than that. So this is an indication that you have already exceeded the maximum tolerated dose, and this is the time to stop further escalation. De-escalate to the previous dose. And so here, the maximum tolerated dose is 20 milligram, and the maximal administered dose is 40 milligram. They may not be same in all cases. Now, let's move to the third question. What should be the study design? It can be open label, single arm, parallel study, or it can be blinded, placebo controlled, randomized in the proportion of 3:1 or 4:1, so that at least 6 to 8 subjects receive the active drug. And the number of subjects can be enrolled per cohort is 6 to 10. This is as per the adverse event label, that it gives in the probability of identifying the adverse event is around 80%, especially the adverse event which are much more common. Multiple cohorts receive higher doses, and the study has to be, the design has to be adaptive, flexible, with plan for dose escalation, de-escalation, provision of interim analysis, as well as, if possible, integrate multiple objectives. And this is a slide showing a typical single ascending dose study with the dosing plan. You can see the first cohort. If the first cohort, the dose is well tolerated, move to the second, move to the third if all the doses are tolerated. And this also indicates the highest dose. If it is tolerated, well, that should be the dose for the fed study, for the food. So this is a typical standard single ascending dose. However, what happens if the dose is not tolerated? That provision is also included at each dose level. What should be the dose for de-escalation? Moreover, if the smallest dose, the first cohort, if they do not tolerate even the first dose, there is a provision of X-1, reduce the dose itself, and even this is not tolerated, specify at what point the drug, the study should be stopped. So this includes each and every aspect, the mechanism, the system for the dose escalation, de-escalation, also at what point of time the study should be terminated. The next, especially with the advent of the concept of the adaptive clinical trial design, this has also been shared in the first-in-man studies. The combo trials, also known as an umbrella trial, that is a combination of single ascending dose with the multiple ascending dose studies in parallel, having multiple objectives. However, start with the single ascending dose in the healthy human volunteers. Once you reach at certain dose level, once you are sure that yes, the drug is well tolerated and you are in a safe zone, in parallel, start multiple ascending dose in patient. And once you reach at the highest tolerated dose, similarly, at the same time, you can also do parallel studies for effect of food, age, gender, and drug-drug interaction. And this whole concept of flexible adaptive trial design, where you can save the time, the cost, and you get the results with a short period of time. It is, explained in this figure. Now, let's move to the next question. What are the spatial requirements to conduct the study? Obviously, if it is done on healthy human volunteers, it can be single center, in-house, at a clinical research organization with phase 1 facility, which is approved by DCGI, or if it is done in hospitals, especially the cytotoxic drugs in cancer patient, even that can be done at the hospital. However, the site should have all the facility for resuscitation measures. In fact, especially in phase 1 unit, there has to be a central monitoring system. In the hospital, you definitely have an intensive care unit and all other facilities. But in phase 1, you need to have a central monitoring system, a sort of an ICU setup. The subjects can be healthy volunteers or patients. It depends on case-to-case basis, your study drug and protocol. And the screening method, especially, you need to screen before you enroll them. It is no different than any other clinical trial. However, there can be protocol-specific tests for the subjects, such as 24-hour ambulatory ECG, pulmonary function test, liver function, renal function, ophthalmological test that has to be done. Quite often, when you're screening the subject, we come across certain situations where there are results are out of range. So what should be done in this situation? For example, the serum transamine is very commonly observed. The results are out of range. RBC in urine, nodal rhythm in ECG. The best is to get assessed, evaluated by physician whether they are clinically relevant. If a subject experts say it is not relevant, there is no harm in enrolling these subjects. However, some monitors or auditors may consider these as a protocol deviation, and that is why this needs to be discussed by your team members before enrolling them. The investigator obviously has to be very qualified, trained, experienced clinical pharmacologist. Ensure the presence of cardiologist, intensivist, study physician, or any other subject required as per protocol or study drug. Yes, a very important aspect of the first-in-man study is safety monitoring and assessment. It has to be done at check-in, check-out, during the whole stay in the CRO, as well as post-study. It has to be complete clinical, biochemical, hematological assessment, continuous monitoring at some fixed point of time as per the protocol, continuous cardiac monitoring, pulse oximeter, even especially post-dosing for certain period of time as per requirement. Now, if the investigational product is to be administered intravenously, it is advisable only to dose one subject at a time. Do not dose all subjects at a time. If it is to be given orally, at least 2 or 3 subjects on day 1, observe them, and then subsequently you can dose. All subjects to remain in clinical facility after a certain period of time post-dosing, and at the time of dosing or before dosing, you can give a questionnaire for adverse event. It has been observed that subjects in a clinical facility, they often have trivial symptoms, which may be related to study drug, which may be because of the environment, in a closed environment with no physical activity, as well as dietary factors. So really, to discriminate this, the questionnaire given before dosing, that will form a baseline, and then subsequently on each day will definitely help you to discriminate between the adverse event or any trivial symptoms which the subjects complain. The follow-up should be at least for 7 to 8 half-lives of the study drug. Give them a diary at the time of checkout. Give them a diary to document the adverse event occurring at home and what was the action taken if required. All subjects with adverse event, with or without adverse event, including the abnormal lab results, should be followed up until it is resolved. A very important aspect of the first-in-man study is the interim review of data. This has to be frequently done jointly by all the team members as well as certain stakeholders to review what is happening, to even decide whether the study should be continued, require some changes, require some additional things. For example, the overall progress, what is happening, the number of subjects, the dosing done, which are the doses which are completed. Tabulate the adverse event, the type, severity, duration, action taken, outcome, causality assessment. The complete table form of the clinical data, the lab data, biochemical, hematological, as well as ECG recording. What is happening to each subject at different point of time? Correlate with the pharmacokinetic data, if possible. That will give you an idea whether there is any relationship with the Cmax or Tmax. Dose proportionality, especially even if there are 3 subjects or 6 subjects at each dose level, that will give an idea about the dose proportionality, whether it is following a linear dose or a steep dose response. And most of the first-in-man study, the oversight is by appointing an independent data safety monitoring board. The endpoints in early phase trial, obviously, is the safety, tolerability, and dose range. To correlate the adverse event with the pharmacokinetic data, identify maximum tolerated dose, and recommend the dose for phase 2 studies. Yes. So what are the different factors for a successful outcome? The general principles remains the same, but the most important is the strong preclinical data, especially novel or MABEL, obtained from the metabolically appropriate and sensitive animal species. That is very, very essential. And follow a conservative and consistent approach to calculate the maximum recommended starting dose, because the priority is the safety. To avoid killing prematurely a drug, which will also kill the team spirit, it is better to start with the safe drug dose, which is equally effective and active. Secondly, a flexible, integrated, intelligent study design, which allows interim analysis, dose escalation and de-escalation, at the same time integrates multiple objectives. Last but not the least, the first-in-man study is not only the investigator, it is the whole team, which is well-trained, a dynamic team with a good coordination and communication. That is, these are certain factors which results into successful outcome. Thank you very much.
Thank you, Dr. Veeda Desai, for nicely covering the different types of study designs, strategies, facilitating factors like planning, flexible protocol, and dosing plans that are indeed very, very necessary for successful execution of phase 1 trials. Now, we move towards the second half of this session, and I would like to welcome our second presenter for today, Dr. Kiran Mathak, who is currently the Head of Medical and Regulatory Affairs at Veeda Clinical Research. He has a rich experience of conducting phase 1 studies with more than 20 studies to his credit, not only in healthy subjects, but also in patients involving many therapeutic segments. Dr. Kiran Mathak is one of the accredited members for GCP training by CDSA India, and he has contributed to the new clinical trial rules implemented in March 2019. Dr. Mathak is also the co-chairman of the medical committee of IDMA and a member of the pharmaceutical and biotech committees of the Confederation of Indian Industries. He's a renowned speaker in international and national conferences, besides being actively involved in academics. Now, I would like to virtually call upon Dr. Mathak to resume this session. Over to you, Dr. Mathak.
Thank you very much, Yuragi, and thank you, Madam, for a wonderful presentation. And good evening to all the participants. Can you hear me, Yuragi?
Yes, yes, sir. We can.
And can you see the slides?
Yes, sir.
Okay. So good evening, everybody. And what I intend to do is for any phase 1 study, if you want to have a successful outcome, what you need to do is the safety of the subjects and how to see that you achieve your target of the safe conduct of the study. So I'll be concentrating more on the safety aspect. And at the same time, I do have 3 or 4 slides of which are more or less similar to Dr. Meera Desai, which I will be skipping. So you have to pardon me for that. But at the same time, I would like to draw your attention, if you, as you see in this slide, the total collaboration of the various stakeholders in the organization. And that starts with formulation scientists, which designs the dosage form and the clinical supply, clinical development scientist who deals with the disease consideration and also the patient characteristics, clinical operations specialist, the toxicologist, clinical pharmacologist, regulatory affairs specialist, and the safety physician. So unless and until all these experts, they put their heads together and do the thinking, wear a thinking cap, learn from the animal studies, and think about the for which indication this drug is to be targeted, and accordingly design the phase 1 study. Also, one has to keep in mind for any new chemical entity development, first thing that is required is to prepare a label claim, because based on that label claim you can decide the kind of activities that you need to carry out. So that is the very important aspect of all the cross-functional collaborators. They have to sit together and design these studies, and particularly the first in human study should ensure that the study design takes into consideration all preclinical learnings on the drug candidates. That is why the preclinical tests are of tremendous importance. And that is where now a new science, which is called as translational medicine, which is coming into picture. And it guides to the various investigators, the doctors, the clinical pharmacologists, the pharmacists, about how to move forward for the successful completion of not only phase 1 study, but also how to move forward for phase 2, phase 3 study. And ultimate goal is to have the NDA submission over a period of time. The most important aspect is, as I said, the safety in phase 1 trials. And the overall safety incidence of serious adverse events with the drug is about 0.02%. Only serious adverse events have been reported. But it's not that there are no serious adverse events, so there are no deaths have taken place. If we see in this, as I mentioned in this example, a man died of cardiac arrest after taking an investigational medicinal product in a trial in Ireland in 1984. And the error that was made by the subject was that he did not declare at the time of screening that he was given a depot injection of an antipsychotic medicine. And we do know that the depot preparation of the antipsychotic medicine can cause prolongation of QTC interval. And that can ultimately lead to death in this subject. And if the subject is particularly of the elderly age group, it can cause still more problems. As Madam mentioned about the study design, there are many factors which are important in designing the study, barring about what Madam mentioned, and that is not only the study design, but also including the study size, the population, the dose escalation scheme specific of the dose limiting toxicity, patient selection, and secondary objectives. Most of the sponsors, they like to have, as Madam mentioned, the combo kind of a study, and it includes the incorporate, they like to have the multiple dose, food effect study, relative bioavailability study, QT prolongation studies, or even a drug-drug interaction studies. Sentinel dosing is already covered, but one of the most important aspect is the EMEA guidelines is very particularly emphasizing that it has to be carried out only in SAD dosing as well as in MAD dosing, uh, as a sentinel dosing only, unless and until you have a very clear scientific rationale not to do so. So it is compulsory actually to have the sentinel dosing, which is a guideline set by EMA guidelines. The dilemma many times is whether one should uh, include healthy subjects or the patients in first in human studies. Now, by and large, the preference is given for the healthy subjects because they are easy to find and also easy to complete the study. At the same time, when there is a comorbidities or concomitant medication being administered to the patient, it is difficult to assess the PK level. There may be drug-drug interaction, or there may be some serious adverse event, and one doesn't know whether it is related to IMP or whether it is related to the concomitant medication the subject is taking. And in some cases, it may be crucial to assess the safety and tolerability of a novel drug candidate prior to testing in compromised patients. But in enrolling the patients, it's very important that particularly where there is a very high risk involved, like cytotoxic drugs or the drugs which are used for HIV, or there are certain life-threatening diseases where all the existing therapeutic options have been exhausted, or if there are any invasive dosing routes. Now, there are certain, particularly what we have seen, even for the bioequivalent studies, and particularly in the biosimilar side, there are drugs which are needed to be injected into the eyes for, it's an intravitreal route, or it may be even intrathecal route, or it may be even directly into the tumor mass. So in such cases, it is always preferred to take use the patients as the subjects. Dose escalation is very important, and it is to be carried out based on the risk-benefit analysis. And if it is not carried out in a proper fashion, one may abandon the study, and which may not be required to be abandoned, and there can be an unnecessary stoppage of the study. This is already covered. Now, there is something which is called as an accelerated titration design, which was, and in this case, it's an accelerated titration design. There is designed by the Simon and his colleagues, and it provides the intra-patient dose escalation until a dose-limiting toxicity is observed. That means within that particular patient, you keep on increasing the dose at a frequent interval depending upon the indication. And once the dose-limiting toxicity is observed, and in that case, the design later on switches to a 3+3 design with 40% increase in the dose increment. So that is another kind of the design that one can also follow, particularly in the oncology indication. There is also a modified continual reassessment method. And in this case, after each dose level, the statistical model predicts a maximum tolerated dose that is higher than the next preset dose level. Then dose escalation is allowed to the next preset dose level. So it is that every group is judged by the statistician or the statistical model, and it sets the MTD or a DLT for the next dose. And this allows more dose levels to be evaluated with a smaller number of patients, and more patients are treated at a close dose, which is more near to the therapeutic dose. And only disadvantage in this is that it does not save time. It's not easily implemented without access to the biostatistician support. So here the biostatistician plays a very major role as such. This is the combo studies, very important, and particularly it is very important where there is a non-linear PK which is expected and also seen in the preclinical study. And particularly the designs where we try to study the food effect study or a formulation effect study, it needs to have a crossover design as opposed to this regular SAD study which Madam showed here, in this, where there is only a single ascending dose study, it is not a crossover design. It is, though it's a double blind when it is administered along with the placebo. But when you start doing the studies where we are trying to compare the gender, we are trying to compare the drug-drug interaction, there is a food effect study, or there is the formulation effect study. In such cases, there is a crossover between the two groups. And the advantage here in this combo design is when you are at the third dose of the SAD study, you can initiate the MAD study direct, straight, and so that you save a lot of time. And in such cases, in this kind of study design, you can save as much as 12 months. And the advantage is there are certain regulatory authorities, they want to have at each stage the regulatory approval. That means you carry out the SAD study, submit the response or the results, and then start the MAD study. You study the MAD results and then go for these studies. And so it takes away a lot of time and also a lot of financial burden on the organization. So in this, one particular protocol can give many results, and also it saves a lot of time, and also it's quite financially quite economical. I have got some of the graphs which will try to explain to you what can happen. And this particular graph I have borrowed from the FDA site. And in this, you will see that there is a population mean for a product. What can happen when you are doing this study? And this is a population curve for a particular product. And based on the study that you carry out, the minimum AUC which is required is this for the efficacy. But if you are doing a study in hepatic impaired patients, now hepatic impaired, renal impaired is also a part of the phase 1 study, which is carried out at a later date when one is very sure of launching the product, or where they are nearing the phase 3 study, though it comes under phase 1 study. So this particular graph, which shows when there is a hepatic impairment, when you are doing the study with hepatic impaired subjects, there are chances that because of the liver playing a very major role in metabolism, there will be a higher level of the drug as such. And this gives an idea about if there is a genetic rapid metabolizers, in that case, the level can be at the lower side. So this kind of information you can derive, and then you can accordingly play a role in designing the subsequent studies. The next graph is the dose proportionality evaluation. It can give you an idea about whether the drug is dose proportional or not. And in this graph, if you see, this is a dose proportional at the various doses, while this one is greater than the dose proportional, and this particular graph shows it is less than dose proportional. So it can also give you this kind of information when you are doing the study. There is also, as you saw in that plan, there is a gender effect. And just last year, I carried out one study with an NCE with the same dose of the subjects, the male cohort, and the another one was the female cohort, based on what we had seen in animal pharmacology, where the female rats and female mice had shown a better PK levels than the male rats and male mice. And we expected that probably it may happen the same in case of human being too. And we were absolutely sure, and what we saw was this kind of result where the PK levels were better in females, particularly the Cmax was better in females than in males. And this resulted into the MAD study that we were planning to do for a female indication in a female subject, where we ultimately reduced the dose by nearly 50% compared to the male subjects for another indication. And we could get the better results even with the lower dose. So this kind of study can be of tremendous importance when we are doing the gender study. Also the food effect study. This is the graph which is of actual study that like the previous study when we carried out. uh, more or less similar uh, uh, kind of a graph that you see here. And the one in the red color is the fasting study in the same cohort who received the fed and also the same dose. So the volunteers were same, the drug was same dose. Only thing is, when in the beginning, in the first time, we gave the drug on fasting, and later on, after about a 1 month of washout period, the same subjects, the same dose, giving the in the fed condition, the Cmax and AUC was higher by 4 times and 5 times respectively. And this resulted into, again, when we did the study as the proof of concept study in patients, we gave the drug at a much lower dose when we started this study in the fed condition. So this was very beneficial to us because it reduced the dose administration, and therefore there were less adverse events also. Even the formulation effect, if you see, the blue graph that you are seeing here is an immediate release formulation. The one in the green is the DR formulation, and that's a delayed release formulation. And this is a prolonged release formulation. Now, this is again with my experience previously with one of the NCE work, phase 1 study when we were doing. What we saw was this was an alpha blocker, and when we did the study with this kind of steep rise in the curve going like this was causing severe giddiness and the postural hypotension in the subjects. And when we tried to study, we saw that all the giddiness or the postural hypotension was during the ascending phase of the curve. And when we discussed with the formulation development colleagues, and we asked them that can they design a prolonged release formulation, more or less maintaining the AUC. And then what we saw when we did the study again after this new formulation, the incidence of the postural hypotension was reduced to a great extent. So this can be of tremendous help when we are doing the formulation study. This is another example. Again, the study which I carried out sometimes a year ago. In the same cohort of the 5 subjects who received active drug, you can see the kind of variation, inter-subject variation between the various subjects. And this could be very difficult, and it can give rise to a lot of difficulty, particularly when we want to escalate the dose. And this requires a very close observations on the subjects so that nothing wrong happens with the subject, particularly related to cardiovascular system. So this is another example of the inter-subject variability, which can cause a lot of issues in dose escalation. So all these studies that we have carried out, the most important aspect that we have to know is the safety pharmacology study. And in this case, the safety pharmacology assays, tests, and models to predict the clinical risk profile of a potential new drug. And what one has to understand from the safety pharmacology is whether there is any variability in extrapolating from animal toxicity studies to studies in human beings. But certain time it may not happen. And what could be the reasons? And that can be due to uncertainties due to enhanced sensitivity in humans compared to animals. Certain toxicity we cannot judge from the animals because some adverse events like headache or myalgia, animals cannot complain. So we cannot judge it. Also difference in the receptors densities and the affinities, unexpected toxicities, interspecies variation in ADME. There can be in animal pharmacology when we study and the safety pharmacology when we study, there is a steep dose response curve. And this can indicate a great risk to the humans because as you could see in the previous graph also, the steep rise can cause a lot of adverse events. The nonlinear pharmacokinetics in the animals will lead to a difficulty in the dose to understand and predict the dose-related toxicity. and the variable bioavailability, as we had seen earlier in the safety pharmacology, and then we also saw in the human beings, and that was the poor bioavailability in test species may underestimate the toxicity in humans. The safety records till now, more or less, if you see, a woman died after receiving a high dose of lignocaine. She was under the endoscopy studies, and she was sent home, and she died at home because of the, as we know, that the lignocaine can cause the effect on the QTc interval. And another woman with mild asthma died of lung damage after inhaling the hexamethonium in a trial in the USA in 2001. This is one of the product that 2006, the monoclonal antibodies, the serious anaphylactic reactions occurred. But one of the important thing happened after this particular tragedy is that in UK, earlier days before 2006, the phase 1 did not require any approval coming from the regulatory authorities. You can take the approval from the ethics committee of the institute where you're conducting the study, and you can then start doing the study. But after this particular incident, the regulatory authorities made it a rule that everybody has to take the approval for conducting the phase 1 study. The consideration in human trials that one has to carry out, and based on the non-clinical studies, and that is to look at what kind of a test drug it is, what is the targeted treatment, the population to be studied, route of administration, and the species in which the study needs to be carried out. Potential target organ toxicity is very important. Pharmacokinetic and pharmacodynamic assessment, safety in special population. Safety considerations are, as I mentioned earlier, is of tremendous importance. And one has to check whether the protocol is well designed, whether the quality of investigational product, that is a GMP batch, has to be there. So the quality aspect of the formulation is very important. I do know one of the person or one of the company came to us and they told us that in the API they had some foreign material. So that is something which one has to keep in mind. The route of and the rate of administration, whether it is appropriate or not. The safety considerations, what is the mode of action, whether it is a novel mechanism of action, what is the nature and intensity of the effect on the specific target. One has to keep in mind, particularly if the drug has got the multiple signaling pathways, which can ultimately lead to or cascade to cytokine release and can cause to severe and serious adverse effect, including the anaphylactic reactions. The dosing part we have already covered, so I will not respond. Dose escalation, it has to be carried out in a proper fashion and as per the scientific guidelines. The number of subjects at each dose, it has to be appropriate so that you know exactly the kind of the effect that is being observed. The duration of the study, it will depend on the particularly the multiple dose and also in the SAD dose. In SAD dose, the subject receives though one single dose, but depending on the half-life of the expected half-life in the human being, one needs to keep the subject in-house and collect the blood samples at the frequent intervals. And based on the half-life which is seen in SAD, the MAD study also needs to have a proper duration within the unit so that you can judge the adverse event profile of the product. Therefore, to judge the safety, we need to carry out the safety monitoring. And for this, you need to have the safety officers or the people who are managing the pharmacovigilance in the organization. You need to collect the data, analyze the data, understand the potential signal of that particular adverse event, and also the limitation of the study which is being carried out. And in these cases, you need to carry out the various vital examination, continuous ECG, hematology, laboratory investigations, also ophthalmologic, psychometric testing, monitoring of suicide scales, radiographic studies. Now, this is, I want to give little more time on this, and that is the cardiac safety monitoring. And in these cases, it is very important, particularly when we are doing the SAD study, to collect the ECG in three copies, and also at every ECG level, collect the blood sample, and it gives you an idea about the concentration response curve. And that will give you an idea in case there is any changes in the ECG at a particular blood level. And that gives you an idea of the concentration response. And if there is no relationship between the concentration and the ECG, you can avoid doing the thorough QT interval study at a later date. Certain dramatic events like anaphylactic shock, Stevens-Johnson syndrome, acute liver failure, they are all related to the drug. There is no doubt about that. The other aspect is to monitor the clinical signs, the duration of clinical observation, the anticipated response to product, and most important aspect is the when to stop the drug administration, when to stop the dose escalation, and when to stop enrollment or totally stop the drug is very important. And these are the various adverse events which can occur, right from the common adverse event to the unexpected adverse events to the serious adverse event. And in this case, therefore, to evaluate the adverse event, there has to be a proper duration of monitoring and also the frequent monitoring at the time of the study which is going on. And one has to understand the laboratory test data and also the limitation of the normal values, at what level one has to consider the particular laboratory test as the serious adverse event, and should one should think of whether one should stop administering the drug to the subject based on the laboratory findings. And dose stopping criteria by EMEA, and this is based on the maximum exposure, and if one reaches the AUC as per the expectation, one can stop conducting the study and stop at that level. If there is any change needs to be made based on the toxicity, it has to be carried out, and one has to generate the rules when to stop based on the list of acceptable toxicity and the procedure for the occurrence of other toxicities. But once you stop the study, let's say there is an adverse event which makes you, as per your plan, to stop the drug. In such cases, what are the action needs to be taken? Hold the subject dosing, understand the toxicity, and then decide further. Evaluate additional subjects, whether one has to particularly continue the subject in the same dose or not. Implementation of smaller dose, if at all, if there is a severe adverse events, maybe you can reduce the dose and continue with the study, or exclusion of certain subjects which are more at a risk for developing the adverse event. One of the important aspects which every regulatory authority appreciate is having the data safety monitoring board, which is also known as DSMB, for every study which is carried out. And this DSMB is governed by the rules, and also it decides based on the safety pharmacology, and based on the protocol, they decide about when the study needs to be stopped, when the dose can be accelerated, or whether the dose can be reduced based on the findings. So DSMB plays a very major role in every cohort of the SAD study and MAD studies. Predictable toxicity, one has to predict the toxicity. I think I will avoid this in the interest of time. But certain times the adverse events can be beneficial. As I had shown earlier, there was a postural hypotension. When I was doing a study on alpha blockers, we could actually make out who is receiving the active drug and who is receiving the placebo, because most of the subjects who were receiving active drug were having a postural hypotension. So there is an adverse event, but also it indicates that the drug is acting. And also another study that I was doing a few years ago on anti-diabetic drug. And again, we could make out when we were doing the frequent sugar levels that which subject is receiving an anti-diabetic drug and which subject is receiving placebo. And thanks to the reduction in the sugar level, but again, it indicates that the drug is acting. So there are certain pitfalls in phase 1 studies, particularly the chronic toxicity cannot be assessed, it cannot be identified, and uncommon toxicities can be missed as such. The biomarkers plays a very major role in first in human studies based on the product and based on the indication. One should develop the biomarkers as such, and that gives a very good idea about the efficacy and the safety of the molecule, and it gives you an idea about whether you should move forward or whether you should stop the study. Immunogenicity plays a very major role, particularly when you're dealing with the biosimilars. The last 1 or 2 slides, and that is challenges in the phase 1 studies. The subjects, healthy or patient, who volunteer for phase 1 trials get no potential therapeutic benefit from the NCE. The risk of harming the subject should be minimal. The risk must be assessed before each trial, especially during the transition from preclinical studies to the first trial. The uncertainty about the tolerability and safety of the IMP is usually at its highest before we start the first in human study. The sponsor must have the clinical data reviewed by the people who have the appropriate technical, scientific, and clinical expertise. At least one reviewer should be independent, and sponsors who do not have the expertise themselves must use the external advisors if required. So in conclusion, before every first phase 1 trial, as well as assessing the risk and justifying that assessment, there must be a strategy for ensuring that any risk is minimized throughout the trial. The sponsor must give them an access to people with responsibility for the relevant preclinical work. The sponsor's physician should liaise with the investigator. The investigator still have concerns about preclinical data, then they should consult an expert. Assessment and management of risk should be documented. The strategy for managing risk should be considered at all aspects of the trial. Correct method, correct population, and dosage selection is of utmost importance. Phase 1 ultimately paves the path for the POC study and phase 2 study. There are other phase 1 studies like renal impairment subject study, hepatic impairment subject study, drug-drug interaction, cardiac toxicity study. These all are, though they come in the phase 1 studies, but they need to be carried out at a later date based on the progress of the various clinical trials. And one of the most important is that highly knowledgeable with analytical mind and good experience medical doctors team plays a very major role in the whole conduct of the first in human being study as such. And thank you very much, ladies and gentlemen, for your patient hearing.
Thank you, Dr. Kiran Mathur, for nicely covering the most crucial aspect of phase 1 clinical trials, the subject safety and various safety designs, and also the things to keep in mind like the population in which the drug will be used, importance of safety monitoring, safety stopping rules, and much more. I'd like to take a pause over here. We understand that this session has kept us all well engaged throughout, but due to unavoidable time constraints, we would not be able to take any questions at this point of time. But please be rest assured that we will be sending the answers to each and every question of yours via emails. Also, we will be sending across the recorded link of this webinar on all the registered email IDs. Lastly, on behalf of Veeda Clinical Research, I would like to sincerely thank you all for being here today and taking out the time to be a part of this webinar. I would also like to extend our gratitude to Dr. Meera and Dr. Kiran for being such wonderful, wonderful presenters and give us all some great learnings today. We at Veeda Clinical Research earnestly look forward to host you all again. Till then, take care everyone, and thank you everyone once again.